So good afternoon, everyone. First, I want to thank the organizer to give me this opportunity to share our research here. So I'm Z from Cincinnati Children's Hospital. I'm a PhD candidate from Dr. Maradi and Dr. Miski lab. Today, I'm going to share our research on single-cell resolved chromatin accessibility maps of inflammatory bowel disease, intestinal, nominate cell types and contexts, mediating genetic disease risk. To get you started, a little bit background about the inflammatory bowel disease. It is a group of chronic disease that cause inflammation of the digestive tract. It affects nearly 3 million Americans, and there are two major subtypes of it. One is the ulcerative colitis, characterized by continuously inflammation of colon. Another format is Crohn's disease, with patchier inflammation that can occur anywhere in the digestive tract, but mainly in the colon and small intestinal. There is rising incidence of the disease in the pediatric population. However, the only approved pediatric biologics are the anti-TNF alpha therapy. Only 1/3 of the patients actually responds to the therapy and achieve mucosal healing. With that being said, new therapies are needed in this domain to improve patient outcome. So to understand the disease mechanism and find better treatment, we decided to go ahead using the multi-omic approach, which simultaneously measures gene expression and chromatin accessibility from single nucleus. It is a powerful tool to identify cell type and regulatory mechanism in the disease and also patient response to the therapy. Those can be done through building gene regulatory network and cellular crosstalk network to understand the mechanism. Furthermore, even though the etiology of IBD has not been fully understood, genetics has known to play a very important role in the disease. There are identified over 400 independent risk loci from previously GWAS study, and most of those are actually in the non-coding region and enriching the enhancers. The mechanism of those non-coding variants has been poorly understood. If causal, they likely can affect the transcription factor binding and regulate gene expression. As we know, ataxic correlates with the transcription factor binding, even though we don't have direct gypsic data from the biopsy sample, but this multi-omic approach can provide evidence for the TF binding. So our goal here is to identify cell types and conditions where the genetic variants likely contribute to the disease, and also the different patient response to the therapy. So with that said, here is our study design. Our collaborators collected pediatric intestinal biopsy from those patients as Crohn's disease and also ulcerative colitis from both ileum and rectum. And there are groups of patients who respond to the therapy show as non-inflamed based on the clinical endoscopy assessment. And also groups of patients who not respond to the therapy show as inflamed. So here on the right is showing a UMAP for all the 319K nuclear with high quality ataxic data that recover from this study. As you can see, there are over 25 different cell types spanning from those lymphocytes, myeloid cell, stromal cell, and also the different epithelial cell population. So to understand the genetics co-localization with the accessible region, we apply this regulatory element locus intersection algorithm. The input to this is a collected GWAS SNP, which are ancestry-based LD expanded, and then we intersect with a cell type resolved accessible region, and then do the randomly shuffling thousands of times to get the non-distribution where we can estimate the significant level. So apply this to our data set, here showing some of the results. In this heat map showing each of the row, it's a collection of the GWAS risk loci, and each column here is a cell type context-specific chromatin accessibility map. And we're showing the relative enrichment across those different cell types in regards to a pediatric PBMC control. There are certain patterns here, such as on the top cluster here, we see an enrichment in particularly Treg and T follicle helper cell population. Those are some autoimmune tracks, such as autoimmune disease and also the PSC, and including the type 1 diabetes. And follow that, we see a cluster of the disease risk loci that are enriched across the immunocell population, including IBD. Bottom there, such as colorectal cancer, those are the diseases where the risk loci mainly correlated, co-localized with the fibroblast and the epithelial cell accessible region. So zoom into this heat map. We're looking at those IBD risk loci, particularly CD4 T cell and macrophage of the inflamed rectum seems to have the strongest IBD risk co-localization. So from there, we decided to zoom in, particularly looking at the macrophage population here. We identified those 5 distinct subpopulations, including resident macrophage, different inflammatory macrophage, and also the inflammatory monocyte population based on some curated marker from literature. Further, when we look at the genome scale comparison, the differential expressed gene and accessible region here shows some shared and distinct pattern across those cell subtype. To look at the composition changes of those subtype, here we particularly look into the Crohn's disease rectum, where we have the most abundant data to draw the conclusion. We can see there is a composition reduction of the resident macrophage under tissue inflamed context, and then the increased composition of inflammatory macrophage and monocytes. And similar trends we found in the ulcerative colitis rectum sample. So with that, we want to further understand what particular subtype actually play more important role here in terms of the genetic predisposition. So here we cluster those differential accessible region into mainly 5 clusters, which are relatively more accessible in one or a few cell type, as shown here. And then we tested the rely enrichment for those cluster of the peaks for the IBD risk loci. Here we can find for the accessible region more in the inflammatory macrophage population, particularly the Crohn's disease risk loci are enriched. And then the inflammatory monocytes high accessible region are co-localized across the Crohn's disease and the ulcerative colitis. So starting from, there was some issue with the display. So what we actually doing is starting from those risk loci, using some of the available EQTL data and also from the activity by contact model data to infer the potential enhancer targets within the inflammatory monocyte population. And then we compare those to the gene that are differentially expressed in this particular cell type. From there, we find a list of the gene that's supposedly to be shown here. And what I want to point it out here are 2 transcription factors. One is the IRF1, the other is the FOS. And FOS particularly are differentially upregulated in the inflammatory monocyte. So we decided to look at the FOS activity. And here is showing an IGV browser kind of a shot to see the IBD risk loci that is predicted to regulate the FOS expression. And here we apply this max attack model to generate in silico TF chip-seq for FOS. The idea here is that we have those pre-trained deep neural network model that takes DNA sequence and a taxic profile as an input, where they can predict for those transcription factor binding sites in a cell type specific manner. Here on the right, I'm showing you near the TNF AIP3 region, here are some FOS binding sites across different cell types. And particularly for those set of the peak, we're testing the enrichment for the false binding compared to the background. And particularly, we find those peak regions that are relatively high accessible in the inflammatory monocyte, they have like a high enrichment of the false binding with high odds ratio, particularly in the inflammatory monocyte compared to other cell type. And then when we look at the false binding site proximal gene, there are actually many enhancer targets of other IBD risk loci that are showing here. And when we do the enrichment analysis for those genes, we find the top enriched pathway are actually the TNF alpha signaling through the NF-kappa B pathway, suggesting FOS itself as an IBD risk loci enhancer target could affect and regulate the TNF signaling. With that, we have some ongoing work trying to connect those genetic risk variants to the transcription factor binding and also cellular crosstalk and gene regulatory network. And finally, I want to thank all the Mirati Lab members, particularly Dr. Wiman, who co-lead the project together with me, and also Akshata, who helped with running MaxAttack for TF binding prediction. And thanks all the collaborators who contribute to the project and the funding source. And lastly, we have this early version preprint on the archive for this study. And I'm ready to take any question. Thank you. If not, I'm fine.
You mentioned the key regulated one was post-transcription factor. Can you elaborate its function on functional state of different macrophages in monocytes?
Yeah, sure. So to my current knowledge, that here, as I showed, it actually regulates the TNF alpha signaling. And also what I know is that for one important regulator, the ETS2, it also work together with FOS to regulate some of the key pathway in the macrophage population. But in terms of their genes regulated, particularly in the inflamed monocytes compared to the inflammatory macrophage, those genes, they're actually enriched for different pathway.